Wow, I've never had such a hard act to follow, I think. So this will be a slight change of pace. Going from, I'm going to stand up actually. So going from probably the highest level of learning from nature in terms of how we organize our culture, down to the smallest level of how we might mimic nature in terms of the smallest thing you could say might have an individual structure, a single molecule. So this is a big old step, but hopefully I can bring everyone with me. So my name's Patrick Parkinson. I'm a lecturer at the University of Manchester. I'm a physicist, and I've tended to work with chemists. And my interest is really in investigating what I would call novel materials, so new materials for optoelectronic applications. And these are applications like LEDs for high-efficiency lighting, photovoltaics for solar cells, or other things like nano lasers. I'm interested in a whole variety of different kind of materials, but this talk is going to focus on molecular materials. So molecular materials are highly promising for a whole variety of these optoelectronic systems, and one of the particular reasons is the ability we have to make small changes to molecules to change their functionality, coupled with the idea of chemistry that can be expanded out and make enormous amounts of a material if it proves to be the most important one. So I will be talking about molecules for photovoltaics. Now, if you went and asked, well, almost anyone, what's the most widespread, efficient and robust form of taking energy from sunlight, people would rightly say photosynthesis. So this is maybe a clear first idea that you might have, that if you wanted to try and understand the tricks that nature plays to make highly efficient, to extract light, to take light and make highly efficient chemical energy to power plants and bacteria, investigating photosynthesis might be a really positive way to do this. And so it seems like a reasonable first step. Let's go and investigate photosynthesis, look at the chemical tricks that nature's played and has developed through evolution, and try and work out what we can take from that to develop our own molecules or our own photovoltaic systems. So when you put it like that, it sounds pretty simple. Unfortunately, it hasn't really been so easy to do that. And there are a number of reasons. So through this talk, effectively I'm going to introduce from the first observation of nature and how that was able to, how nature has developed ways of taking solar energy, onto how scientists have then gone on to understand how it works, to really try and identify what features of photosynthesis give us high efficiency, and through to more recent work on mimicking. So what can we take from nature? What can we, I hesitate to use the word exploit, but it is exploit the development that has been done by nature to make more efficient devices ourselves. So all of this is underpinned by a single development, so it's worth just introducing this, although this is not the core part of the talk. I'm talking about solar cells. So I'm talking about things that we can generate electrical energy. This is what we want in modern life. Photosynthesis in general produces chemical energy, so there's a little bit of a disconnect here. But the enabling technology behind generating energy from molecules, one of the enabling technologies was described in this really rather seminal paper by Brian O'Regan and Michael Gretzel back in 1991. And this was the development of this technique known as dye-sensitized solar cells, which is effectively a device made of simple things that, as engineers or people who are involved in electrical materials, are very familiar with. Metals, semiconductors, flows of electrons, and we can power our normal electronic devices using a solar cell made of this structure. The interesting thing about this structure in particular is this bit here. It's called the dye. So there is a step. And this dye is particularly interesting because it allows us, in this device, we're able to separate these two processes. One, the absorption of light, and the generation of an excitation, which is energy stored in some kind of electrical form on a molecule. And we're separating that from the next step, which is taking that electrical energy or that excitation energy on a molecule and using it to power a device. So I'm going to be focusing on the molecules, but this is a potential way that you could then, once you've developed a molecule and addressed several other challenges, you could then get electrical energy from it. And so, as I say, this is a really interesting paper, a really seminal paper. It's about 25 years old. It's about 22,000 citations now. And if you go online, you can actually make these things really easily and make them in school. And changing the molecule gives you a load of different properties, so you can go from things that are very cheap and easy to make, or to things that are very complicated and hard to make, changing things like efficiency, the colour of the solar cell. There's a lot of interest in this. And one of the most interesting that I like is you can, in fact, just squish blackcurrants and get the juice out of it, and that will give you photovoltaic performance. So you go online and you look for dye-sensitized solar cells. There's a little sheet on making these entire things at room temperature with pastes that look like toothpaste, and really nothing more complicated than a razor blade and a blackcurrant to make a solar cell. It only lasts about 3 hours, though. It's not particularly useful. So this is exploiting nature. So I've ticked that box. That's good. So mimicking nature. The rest of the talk will be talking about this, or rather learning from nature. So photosynthesis. Now, I had to go back, given the topic of this talk, I actually went back and tried to understand a little bit more about the origin of when people first started to investigate this. And it comes back to this paper, an amazingly titled paper in 1779, Experiments Upon Vegetables, which I will come back to in a couple of slides, by John Ingenhouse, who's famed for being the first person to immunize a royal, among other things. So in this incredible experiment, which I'll touch on later, which was the first one to identify the role of light in whatever process occurred inside plants that give it energy and create oxygen from carbon dioxide. So that was the first. So it really opened this field of, studying photosynthesis. So that's the 200 years in there. The next important step, at least from my point of view, was really understanding on a molecular level of what actually is the molecule that's giving us, or giving plants or vegetables, this property of being able to generate energy to power itself from light. And so a really cool paper in 1995 used some really complicated techniques to actually draw the molecule, to actually understand it. This is what the molecule looks like, and I'll come back to this a little bit later. Now, being able to understand it was the next step, and then we're really able to accelerate the pace of mimicking it. So we want to understand what the features are of this molecule that give it its properties, but also how can we take those features and adapt them for our own use. So many groups have looked into this. This is a paper by a group that I've collaborated with in Oxford Chemistry, where they're able to mimic these kind of circular structures of molecules and make them of all sorts of different sizes, and this is scanning tunneling microscopy. So if we go back to the beginning, let's look at the observation. Energy harvesting from light. Where do we start? Well, the important thing, as many of you will know from high school or secondary school, is when you know about photosynthesis, you know two things about it. It generates energy, chemical energy for the plant to use, but also it converts carbon dioxide to oxygen. This is as kind of a byproduct, or, as John Ingenhouse said, the production of dephlogisticated air, which I also do love. So that's great, and this is a really important process, and we love this, but when we're making solar cells, we don't always want to have to flow gas into them. So really, this was our first stumbling block. We want a molecule that can give us light to electrical energy or excitation on the molecule, but we don't really want to have to feed gases in it. And so that's a big jump for us to start trying to tweak a molecule and try and get it to do what we want to do. But we were lucky, because in about the 60s, if we want electrical energy, not oxygen, it turns out that nature uses photosynthesis in a very, very wide variety of applications, not just in plants, in fact in bacteria. And in 1969, this bacteria, Rhodopseudomonas acidophilia, was found, which lives at the bottom of lakes across North America, actually. And I think some of these were taken from Canada, in fact. And so this bacteria is known as a purple photosynthetic bacteria. It uses the photosynthetic process to create energy to run it, but it doesn't require this carbon dioxide to oxygen process in it. So the first studies that were done on this were literally getting this bacteria and looking at the absorption spectrum of it. And this is a figure taken from the paper. And the thing that you can identify here is the bit on the left, which is the high energy side, which is basically non-functional junk that I'm not interested in. The interesting part is the bit over here on the right. And this is the absorption spectrum of the molecule that's active in creating this energy from light. So the two interesting features about this is, firstly, well, you can't see on this figure because it's not reproduced very well, but the absorption peak of this is around 850 nanometers. So that's in the red. So it's not what we'd normally think of a photosynthesis of the green plants. This is red photosynthesis, which is actually really useful because there's a theoretical study to show that the ideal absorption energy for a material to give us high efficiency in photovoltaics is further towards the red. So that's positive. Secondly, we see two peaks on here. So it gave us an idea, or the first idea, that probably there were two things going on inside here. Maybe two molecules, maybe something else like this. But that's not enough for us to start copying it. or really understanding it to any extent. And it really was the development of the technique, this is actually synchrotron X-ray protein crystallography, basically to say probably one of the most important and difficult techniques that exists for understanding proteins and molecules in the world today. And the actual structure of one of the important proteins, this is called light harvesting complex 2 from this bacteria chlorophyll, from this bacteria, was determined. So this is it with all the joys. So the thing to point out on this one is that the size going across there is about 2 nanometers. So that's a kind of a length scale. Now that's everything that goes on inside there. You can strip away the supporting proteins. These are inactive, inert matrix that just hold everything together. If you strip that away, once you know what the structure is, you end up with something looking like this. So this is the structure that nature has developed, or evolution has developed, to get efficient and robust capture of light and transfer to energy. And so our question is why? This is a complicated structure. And why has this been chosen? What aspects of this are important? So we can, I'm just going to point out 3 specific features that we describe as bacteria chlorophyll B850, bacteria chlorophyll B800, and a carotenoid. You can kind of ignore the carotenoid for the rest of this talk. So these molecules are actually porphyrins, which were mentioned yesterday, and they are very commonly used throughout nature to absorb light. So they're very strongly coloured. So just from a first look, what could be happening here? Well, we see 2 important features about this molecule. One, we have these 2 species, these bacteria chlorophyll B800 and a B850. Why might that be the case? Well, it turns out that porphyrins absorb light. If you hold a porphyrin, this kind of you can't really see it so easily here, but they're kind of a square molecule. If you hold a square, they absorb light this way and they absorb light this way, if it's polarized this way or this way, but not going across it. So if we really want to get omnidirectional absorption of light, we need to have these porphyrins in every possible orientation. So having this structure of the B800's flat and the B850's vertical allows us to get this omnidirectional absorption of light. But a second feature that's really interesting is this overall circular structure. Now the overall circular structure is actually quite hard to make, and synthetically it's very difficult to reproduce. So you feel there must be a reason that nature has developed such a complicated and difficult structure, difficult to reproduce structure. There must be some benefit in the function of this material. So once the structure had been determined, the next step would be for theoreticians to do kind of quantum chemistry and quite hard calculations to work out how energy would move around inside one of these molecules. So there's a paper from 1998 where they did these calculations, very complex calculations of how energy hops between various parts of this molecule, but can be summarized something like this. We have these 3 components in this light harvesting complex 2, bacteria chlorophyll, and carotenoid. If we absorb light in one of those, the energy can hop around between it. It can move around, which is good, because then that gives us, it doesn't matter where we absorb the light, it will end up, in the end, on this ring structure, which is the lowest energy part of the molecule. But after it's finished on this LH2, we've got an excitation on this molecule, but it's got nowhere to go. So in a real membrane inside one of these bacteria, it can then hop onto another LH2 molecule, and eventually hop onto what's known as an LH1 molecule, where the chemistry occurs, or at the reaction center, where the actual conversion of this light energy into chemical energy. Now that's the bit we don't care about. We don't want chemical energy, we want electrical energy. So we really need to understand this light harvesting complex 2 and not really the other parts. So I've mentioned that having this B800 and B850 in different orientations gives us this omnidirectional absorption. But what about the ring? Well, the ring structure is actually really cool. It turns out that quantum mechanics plays a significant role in these molecules. And the way that it plays a role is something known as delocalization. Now, for those who may be not familiar with quantum mechanics, this is the idea that you can't really determine precisely where excitations are on the very smallest length scales. They're delocalized around the entire molecule, at least on some time scale. So when we transfer energy onto this ring, it exists everywhere on the ring. And then it will localize due to thermal processes, or it will go towards an energy minimum. Now, the reason this is interesting is if you stack a whole load of rings together and you want energy to transfer between them, this delocalization effect gives you the best opportunity to hop onto whatever the next site is, because each time you hop, it delocalizes, and you hop onto the next one, you can get delocalization, something like this. Now, this obviously, I'm not a quantum physicist, so this is a kind of a side effect for me, but this prompted a huge amount of research over the past 10 years. One of the papers showing evidence for wave-like energy transfer through quantum coherence in photosynthetic systems. And slightly more recently, this other paper showing that kind of quantum coherent effects can occur even after energy is transferred from this antenna to the ring. Not too important, but I just wanted to bring up these two slides because they prompted a rather interesting paper page on BBC News, which is great, plants, and I'm seen doing quantum physics. Now, the interesting use of those quote marks is, this molecule is seen doing quantum physics, but it is not a plant. This comes from bacteria. So they've managed to completely misinterpret, while writing the right words, completely get the wrong point of the paper. But anyway, there you go. So the joys of the press. Right, so that's, hopefully I've shown you some idea of how these molecules work and how they might be interesting. But that isn't mimicking nature, that's understanding nature. What we want to do is we want to develop new molecules that work to give us high efficiency. That's what we want. And to do that, what we kind of need to do is work out how important the various features, for instance, the ring structure, or this antenna and the ring together, how important these are to the overall performance and robustness of its performance. Because then when we steal from nature, we are going to adapt what has been developed by nature. We're going to need to know what bit to steal and what bit that we don't have to worry about. So ideally, what we'd want to do, as a scientist, what I'd want to do is make tweaks, make little changes, and see what happens. So can we change the chemistry? Can we change the metal inside it? Can we change the structure, the size of the ring, or the number of units in it? Can I change the antenna? Can I put extra ones on there, take some away? Now, it turns out that's hard in biology. You can do gene editing and then try and see what comes out. It has been done. It's incredible research, but for me, this sounds a very challenging way of understanding what's going on. So a better way would be just to mimic the whole structure, just to copy it as a synthetic chemist. Now, I can say this is a better way because I'm not a synthetic chemist. My collaborators were, and I've never seen someone work in the lab for 6 months to produce a microgram of a chemical, and then I just go and blast it with lasers. And so it's not easy, but I can say it's easy because it wasn't me. So the tool that has been used to reproducing this is hard. It's a big structure. It's got loads of components to it. If anyone is a synthetic chemist, they'll probably be aware that this is very difficult to make. The trick is known as supra-molecular chemistry. I just want to do one slide just to say what that is and how we're going to use it to make these, to make our mimics of nature. So supra-molecular chemistry is the use of non-covalent bonding, or hydrogen bonding, to attach two molecules to get a new molecule with greater functionality. So they're not covalently attached. You can think of this as occurring in something like an enzyme you might remember from school, with a kind of a lock and key. One molecule is designed to fit another molecule, and they will go together. That's thermodynamically driven, so they will want to attach together. And the two ways we can use this to do our mimicking of nature are known as template-directed synthesis. In this one, we get the active porphyrin, and trying to form it into a circle is hard. This is a tough challenge. So what we do is we use a template that is circular, and we get them to hop onto the template. It may be clear from this figure. And then we seal the ring, we close the ring, and then we knock the templates out. And that gives us our ring. So that's the first use of supramolecular chemistry. The second use is complexation. So we use exactly the same trick. We get these two molecules now, we get our ring, and we get our antenna, and we use supramolecular chemistry to drive the formation of structures that look like this. They want to attach together, and we can make this. So this is a synthetic molecule that I've studied a couple of years ago now, that is our synthetic analog of natural light harvesting systems. So for those of you maybe more interested in chemistry, this is what the structure looks like. These are zinc porphyrin macrocycles attached with butadiene bridges. So this is covalently bonded around the edge, and we can get decent delocalization. But the important thing is the size. This is about 2.4 nanometers across. So we're making something of a very similar size to nature. At least this was one of the first things. Okay. So this was the first step. The next step was to see how we could then, what can we do? What can we change? And how can we study this? And the most important thing to study, we found, was changing the size. So through the next few years, my collaborators at Harry Anderson and University of Oxford developed this technique to make 6-membered rings, to make 12-membered rings, and then more recently to make 30 and 40-membered rings. Now, these are pretty big, actually. These are huge molecules, there's 600 carbons in them. But they allow us now to make rings that are all the same, but of different sizes. And this is something that, to get nature to do, would be hard. And now we can start to investigate, well, how important is the size of the ring to some of these properties? For instance, the delocalization of energy around the edge of the ring. So one of the first studies I did on these molecules was taking different ring sizes and studying how energy is delocalized on them using optical techniques. So for small rings, smaller than 10 units, sorry, I should say before I get onto that, natural size rings are made of 9 pairs, so they're 18 porphyrins in this circle. Okay. So for smaller rings of size less than 10, we get the absorption of light, we get complete delocalization, but it stays delocalized. It never goes down onto one point, which would then allow us to hop onto the next ring. So probably not what we want. If we go for rings between 10 and 30 in units, we get this effect. Light is absorbed, it delocalizes, it spreads out across the ring, and then very quickly, really quickly, less than a picosecond, it hops down onto one point on the ring, which is what we see in nature. So that's fine. And if we get huge rings, well, it never delocalizes completely to start with, which is probably what we would expect as we go towards kind of macro-size stuff. And so at least in this case, it turns out that the sweet spot is somewhere between 10 and 30. Nature's chosen 18. Okay, the molecules aren't identical, but they're very similar. So there's probably, this is probably a feature that if we want to exploit nature, we're probably going to have to copy the size. So that's fine. So having determined that, how can we make these full antenna nanoring structures? Well, here's the natural one, just to highlight that again. And as I said, well, yes, we can. We can make these structures with these nanorings, where the ring absorbs at 820 compared to 850 in the natural one. The antenna absorbs at 740 instead of 800 in the natural one. But this is great. Now we can make small chemical tweaks to either the antenna or the nanoring to make something that really doesn't exist in nature. So one example of this was one of the first things we looked at. Here's the one that maybe looks a little bit more like nature, although it's still a bit different at the top. And at the bottom, we made a chemical tweak so that we now get 2 rings. We get twice the absorption, twice the number of these the ring structures. So what do? Well, we do an experiment where we shine light on the antenna, and we look at how much light comes back out of the antenna. Now, if the energy hops onto the ring, then we don't get any light back out. And we're able to just basically look at the emission spectrum of these things. And my core research is looking at how these very, very fast effects. So I do a time resolve measurement. So what you can see here is how much light is coming out of this antenna for the top molecule or the bottom molecule in 2 cases. When the antenna's on its own, you get the red line. Light comes out for a long time. When the antenna's attached to the nanoring, the energy very quickly hops onto the ring. So we don't get any emission from it. And so in the top one, we get this transfer in 1.25 picoseconds. In the bottom, 1.65 picoseconds. In nature, it's about 0.9 picoseconds. So the important thing to note here is the faster you can get energy from the antenna onto the ring, the more efficient the process is. The less likely you are to lose energy from that from the antenna at early stages. So we are now able to surpass nature in one specific measure, which might be useful for developing molecules for photovoltaics. So that's all the kind of core science I wanted to talk about. This is kind of a future. The guys in the chemistry department in Oxford have been making these crazy molecules that really, really don't exist in nature. Tubes of things that are twisted out of shape and all sorts of different shape materials, which are really going to allow us to understand the role of shape and geometry in the performance of these. And also are kind of just quite fun for them, I think, to synthesize. So to summarize what I hope I've managed to speak about, nature does a great job. No one's saying that we're going to change what nature does, but we want to do something slightly different, so we're going to need to make changes to it. To do that, we need to do 2 things. One, can we understand the tricks that nature plays? Can we then adapt these tricks for our own use? Or potentially, can we go further? We don't have the limitations that nature has of having to work in certain environments. So there's no reason we can't do better in very specific cases. So can we adapt this for our own use? With that, I probably should acknowledge some of the students in Oxford and the chemistry department at Oxford who were involved in making these molecules. and the funding. Thank you all.
Those are great presentations. I just wanted to ask Chantal a question, if I can remember it correctly now. So you're talking about, the link to the land, and I was thinking about the diversity of First Nation, Métis, Inuit people, and even non-status First Nations, and how, I think those are really great ideas, especially around food sovereignty. But for some, for some communities, they don't have a link to the land. They won't have land-based, they won't be able to be land-based, even for Métis communities. So I was wondering your thoughts around that, because I was thinking in some of the work that I do, I think more around reclamation of identity, reclaiming traditional roles, as you were talking about, gender. I was thinking about that because I have Anishinaabe ancestry as well, and I was thinking, are there other ways to maybe connect those ideas? Just thinking, a little more broadly about that. Definitely. And thank you so much for a great presentation.
Oh, thank you. Yeah, no, that's a great question, and I think that I have, as a geographer and someone who's really, I'm from Northern Ontario, so my first and most formative experiences have been on Lake Superior. And my own experiences as a person who did not live on reserve, right, as a result of lots of complicated things. Anyway, so I have this unique kind of history, and as I kind of had my experiences learning through university, have come to be an urban person. And suddenly I realized that the ways that I'm going to learn Indigenous knowledge and practice and teach to my kids is very different than, say, my sisters who do live in Northern Ontario and have access to elders and people and that land base. And so I guess this idea of repossession has some assumptions built into it that we may not necessarily have access to land in that same way. But it's okay, because the representations and places where we are going to be creating these new forms of indigeneity is not necessarily the same as in the past. And I think that's something that really needs to be talked about in Canada, because I think we have really old ideas about who Indigenous people are, not just in Canada, but globally. And so I think, obviously, I don't even know what the percentage is of contested land and the ways that treaties are the contested land claim issue is hot. It's a hot topic, right? But for lots of different reasons, we are now a really urbanized population. And it doesn't mean that we don't have room and space to be exploring our Indigenous knowledge systems, but in new ways. And so I think that there's political, economic, cultural ways, and that's those are promising places, but I think it's really it's unexplored. And I think to the detriment, because I think that we find a lot of kids, Indigenous kids, who are living in cities and don't know where their place is. And it's really sad. So that's why I'm hoping that we can explore that a bit more so that everybody knows where they belong.
Okay. Thanks. Yes, I've got a question for Patrick about photosynthesis. So I appreciate that you want to do it better than nature, but shouldn't the first step be to actually mimic nature and actually get some photosynthetic PV? Ever since I've read Ian McEwan's Solar, I guess, why aren't we doing this yet? What's actually stopping us from just grinding up blue currents, as you say, in a massively big scale and actually using it in a proper way?
Okay, I mean, that's a good question. Probably the thing that I didn't mention at all was, you'll see why, was that these molecules that I'm talking about will never be used in real solar cells, okay? Never. They are outstandingly difficult to make. Really, the kind of pinnacle of synthetic chemistry, you can make fractions of grams, milligrams, over month periods. Okay, so it really is a project of understanding at this stage, because we're never going to make these molecules. It's about what do we need to understand about this to make our own ones. So I wouldn't say I glossed over it, but I, you know, the purpose isn't to make something in this project. What you're saying, of course, is, you know, really interesting, and actually the diversity. I mean, it was mentioned in a talk yesterday about the, you know, the diversity of what's required in terms of generating energy from light, from people using pure just bacteria to create chemical energy. And, you know, people do this for, you know, even going to things like hydroponics. You know, you need to define your problem and use the right solution to the right problem. So I think to get back to your kind of specific question, can we just take nature and use it to generate energy? It's not typically, at least in my awareness, it tends not to be so easy to directly extract it, and you do have to make changes. Also, a lot of biological proteins work very well in biological environments or physiological conditions, and that's not normally the same conditions you want to run a solar cell under. No, I hope that.
I also have a question for Patrick. So when you show these rings and you say the words localization and delocalization, all quantum physicists will get really excited and think the thing you should be doing is putting a magnetic field through that ring somehow to turn localization and delocalization on and off. Do you look at this, or does anyone in your community?
So those exact molecules have been studied in this thing, and it's exceedingly difficult.
Okay.
Very fun. Yeah. I can talk to you after. It's one of these things that you would think you would see something really interesting. You actually have to basically put a charge on them permanently. So you have to dope them, and then they only last for a few hours. So you have a chemical problem. And then you have to run them at low temperature, and then you have to put them in a superconducting magnet, and it just becomes this kind of snowball effect of experimental horror.
So I thought I'd try to put a bridging question in here amongst this kind of very divergent, which I appreciate that there's a broad breadth to this panel. But I'm kind of curious about, one of the reasons we really worry about diversity in STEM is that we're worried about diversity of ideas. So ideas where things come from, different use cases that might come out of the technology. We see things like genetically modified foods in different cultures approaching that very differently as to the acceptability of that. And so I'm kind of interested in starting with Chantal, kind of trying to get a sense of how do you see some of these indigenous cultures in their, maybe not education, but their attempts to actually become parts of some of these scientific cultures to bring in those ideas and maybe bring them back or to tie them back in. And then kind of going maybe to the other panel to get a sense of how they feel that's actually happening or not.
I think that's a really great question, and I think it's something that's necessary and important for I think our economies, the global economy really, because I think that Indigenous peoples have been relatively excluded from the scientific community because our sciences are oral for the most part. And you know, the very nature of Indigenous knowledge systems means that we're not going to find a lot about this information in published scientific papers because it's knowledge that people are meant to know when they're meant to know it. So that's one part of the question. So we're talking about different forms of knowledge and the ways that you learn these knowledges, but also because Indigenous peoples have been relatively, I think, marginalized from traditional, and have not had equal access and opportunity to learn, you know, STEM or engineering. Even when our kids get into universities, they're traditionally kind of like pushed into First Nation studies, into social work, to go back to their communities and help in ways that are often crisis-driven. And I mean, there's like this really important revolution, I think, happening through the Canadian Institutes for Health Research, which has wonderfully funded, I think, myself, maybe Carrie, and developed this amazing group of young scholars who are leading the charge on Indigenous health and Indigenous health research. And it's starting to cross boundaries. And I think developing models of learning and success that are not just focused on Native studies, okay? And at Western, one of the very exciting developments has been the creation of this new interdisciplinary initiative in applied Indigenous scholarship. So there are a couple of really important, I think, political contexts for that. The first is that Western's in the process of creating our first ever Indigenous strategic plan, which is awesome. But also because we are trying to respond to the calls to action from the Truth and Reconciliation Commission, which was a multi-year commission into understanding the impacts of the residential school systems, which I talked about. And so there were a number of calls to action that were dedicated specifically for post-secondary educations and for the funding agencies. And part of that was to create more opportunities for Indigenous learners in our institutions and to make them more widely available and accessible and appealing options so that we don't just see, you know, Indigenous kids who get to university and choose these same options, but that they choose engineering and that they choose medical sciences and wide places. And I think there's a real opportunity for all of us to be a part of that so that we can all see a role for ourselves in making our universities much more inclusive places for Indigenous learners and non-Indigenous learners. Because as we push forward in reconciliation in Canada, we're not just talking about making our country better for Indigenous people. It's for everybody, right? As soon as we start to, I think, reduce those inequalities that we see in Indigenous communities, we're all going to benefit. So I think that is a really great question, and I think that promoting interdisciplinarity for Indigenous ideas broadly, there's a real promise there.
Any other questions? Richard?
Did you want to respond?
Sorry, Patrick, did you want to come in on that? One sec.
Only because I feel I ought to. But actually, no, I mean, I find that this is coming from physics, you know. In many ways, physicists tend to think of themselves as having a monoculture. You know, there is one way of doing things and we do it this way, and it actually doesn't matter who you are or where you come from as long as you do it the way we do it. You know, I don't think that's true. That's what we'd like to think. But I was very interested with your answer, and actually to the extent at which we all need to try and understand how much we can gain by aligning the interests to try not to compartmentalize, because I think things tend to work better when we say, well, actually, you know, if we're, as a university, if we take the best people from anywhere, whoever they are, whatever language they speak, whatever culture they're from, we all gain. And whether you can do that without losing some of your culture is something that, I mean, it's outside physics, so I don't know. It's beyond my experience, but I thought it was a very interesting answer.
Okay, so I'm going to put a view forward which isn't necessarily my own, but it relates to something that Patrick's been talking about. It's in photosynthesis. So nature isn't greedy in how it uses photons, right? So a tree will take up to 100 years to grow, and then we can take that tree, we can burn it and get the energy back in a very short amount of time. And what that shows is that it's actually not very efficient. So nature is sort of 3 to 6% efficient for photosynthesis. The best thing nature can do is kind of sugarcane, which is up to about 11% efficient. So if we're going to try and use nature, we're not going to do very well. I mean, we have silicon solar cells, which work at 20% and are much cheaper to produce than a field full of planting trees, for example, here, from an economic point of view. Of course, when a tree grows, it doesn't just create energy and store it for us, it supports a whole ecosystem. Right? And that's valuable in itself as a separate thing. So, two questions. One, is trying to learn from nature in terms of photosynthesis actually going to get us where we need to be for our energy demands as society? B, what about genetic modification? Should we be modifying nature to make it more efficient? Right? This is a controversial question I'm putting out here. And how do we also then deal with the fact that nature's also had millions of years to evolve, and it's kind of got to what you'd think would be an optimum point. So can it get any better, fundamentally? So just some thoughts there for people to try and think about and discuss.
Maybe I could dive in with the first part and then open the hard bit up to others. But you're saying about what can we learn? I mean, is this the right thing to do, to be learning from nature? And I think I kind of made the point a little bit earlier that you're absolutely right. Nature is very, very good at doing what it wants to do. That doesn't I mean, that's not what we want it to do. In a way, I do feel, although I have couched all my work in terms of photosynthesis, you can probably imagine it's to an extent, it is fundamental research about understanding. And I appreciate the point you make that actually we would have a long way to go if we tried to really adapt. You know, we can do better using entirely synthetic silicon cells, or many of the other techniques that have been spoken about yesterday. In terms of, I mean, your second question was, should we modify nature?
Yeah, so nature is, we know it's quite inefficient. We have these great tools now. We have genetic modification, etc. As mentioned yesterday. Where are we as a society in terms of, if we know we can improve the efficiency of nature at growing, which might therefore make crop yields higher? Is that something? So that's just converting energy into another source. Is that something we should be doing as a society? How do we address that issue? It's a more general discussion.
Which one of you want to take that one on?
Well, I think when it comes to energy production and light harvesting in particular, and we'll leave genetic modification for a second, but I think there's more to it than just raw internal quantum yields and stuff like this, right? And there's the ideas around product cycle, so cradle to grave analogies, recycling the materials, green chemistry. There's all these other things rather that play an important role, I think, on the justification between one type of device versus another. And of course, application plays into that as well. But so in some cases, if you can get something really cheap that only has 6% or 11% efficiency versus something that they put into outer space that reuses rare earth metals that cost $1 billion for that much, I think there's some play between those two that can be found. And engineers should probably get into that, I guess. With respects to genetic modification, and I guess this sort of comes back to food as well. I was raised on a farm, and so my grandfather built his cattle herd literally from his dad, right? And his son took it over. That cattle herd was around for 100 years, roughly. And they selectively bred cows, right? They actually genetically modified their food and other things. So I'm not against genetic modification. The problem, I guess, with genetic modification that we should be worried about is the unintended consequences. So, and when it comes to genetically modifying bacteria or cyanobacteria to increase their light harvesting capabilities or to try to disrupt and maybe optimize light harvesting abilities, will we create a super cyanobacteria that takes over the world and kills us all and becomes the dominant species? I don't know, but that's my answer from a bioengineer's perspective. Thank you guys, by the way.
I mean, one of the things is I thought about during these talks is how we're mimicking nature ourselves, but how much does nature respond to the impact of human technological advances? So, there's a two-way process here. So, as humans as we've gone on and pushed the technological boundary, I mean, how much is nature sort of starting to mimic us in certain ways? I know it's very difficult to measure many things over short length times, but do you know if there's any? Well, this is just me talking to myself. I'm not sure if anyone else will.
So I guess maybe what I'm getting from what your question is these microbes do more than just harvest light, right? They store energy in chemical form. They obviously live, they replicate, et cetera, et cetera. And so I know like people are trying to use them as carbon capture mechanisms and things like that, if that's what you're driving at. So there is sort of a feedback between, you know, pollution and growth of molecule or of elements.
I think one of the things is, you know, we sort of put infrastructure in places that are now seeing colonization of species onto them, you know, that aren't naturally found in their environments. So how they're sort of, how they're reacting back to what we impact on them. That was the sort of point I was trying to get at. But again, it's just something that's going around in my head and everyone's looking at me blankly. So I will keep passing the box around. Do you want to just take it down there? Yep. Catch.
Okay, so this might be controversial, and also I'm not a scientist, I'm a humanist. But something that's striking me, I thought was great, the question about what the bridging question between the papers, but something sort of fundamental is striking me, which is this discussion about us and nature, as though we weren't nature. And I think Chantal's presentation sort of underscores the fact that Indigenous knowledge understands us as nature, or as a, you know, playing an integral role in nature or being deeply connected to it. And so I guess I just wanted to hear people's thoughts about that sort of bifurcation between humans as sort of separate, the masters of all, the center of the universe, and consciousness and reason and all of these things, which I believe in, but also understand that old sort of Cartesian notion has been challenged, but also needs to be challenged, I think. That would be my claim. And I think, so when we talk about Indigenous knowledge, it's not just bringing Indigenous kids into the academy and teaching them how to be scientists. It's about scientists going to Indigenous communities and learning from Indigenous knowledge. So I just wondered if people had thoughts about that.
Yeah, who wants to? Patrick?
I completely agree with your question. And actually, I realized through my talk that the language I would use about nature, you know, take, learn, or not even learn, take, mimic, steal, exploit. Right, that's the language that is used. And it's not, okay, I'm not saying, you know, we're going to kill a load of animals and grind them up and stuff, but it is, there is a certain exploitative nature to looking at what nature does well and taking one aspect of that for something we want without a realization, and the point has been made that actually, you know, trees grow slowly for a reason. If trees were 100% efficient, you know, they'd grow in hardly any time, and they don't. And that isn't a question that I'm asking. I'm asking what's the best we can do. So, I mean, I'd certainly agree that it's not something that's rewarded necessarily in physics, my own field, to think about that. And it's certainly not something that if you said to, you know, in a grant proposal, I want to do something that's more in tune with nature and less focused on absolute efficiency, it would be possibly a challenge to get that funded. Possibly, I couldn't say.
I think one of the obviously one of the big challenges is that we are academics, right? And our you know, precisely what you're pointing out is that we are in this system where we've got to produce really quickly, and unfortunately, you know, there's not often time to think about things that aren't directly related to your discipline. And I think what Carrie was trying to talk about yesterday, well, what you did talk about, is, you know, the nature and role of communities and Indigenous communities in guiding research. So this is one like really new and exciting area, I think, of research happening in Canada that is gaining recognition and is going to gain, I think, a lot more recognition post, you know, TRC and so on, by the federal granting agencies, is the role and responsibility for us to be thinking about the human dimensions of the work that we do, right? And that placing Indigenous communities, for example, in research is very important. So we held a panel last week and talked about our positionalities. So it was an interdisciplinary panel, and we're talking about the role for the disciplines in Indigenous research. and we had a biologist on board. And he's amazing because he's the sort of guy who's very interested in mercury in the environment, but is completely guided by his community partners. And unfortunately for him, his he's working in NSERC and the Natural Sciences and Engineering's research, right? Which doesn't necessarily allow for the development of community relationships within a 5-year research plan, right? So we're often not afforded the ability to do those sorts of things so that we can accurately incorporate human dimensions into the applied nature of the work that we're doing. And I mean, the work that you all talked about yesterday, I was just sitting there and so confused, but also enlightened, because I'm thinking about, the pharmaceutical industry and thinking about mice and pushing buttons and thinking about how does this relate into everyday contexts. And it all has important applications, but oftentimes we don't have the opportunities, I think, to bring this back to that human level. And I don't really know how we're going to get there, but I think it's one of the big challenges for us is to learn how to disseminate and not just, it shouldn't happen just at the end of the research, but at the beginning, right? So how are we doing stuff that actually matters for overcoming the big challenges that we see?
I guess, if you don't mind, I'd like to
No, please.
Yeah, I was challenged with this. I was a scientific advisor to curriculum development for K-12 in Alberta. And this idea of being able to communicate, like you were saying, Patrick, really came up. We were building basically mini labs, nanotech labs that kids could do in school to illustrate some different phenomena. And anyway, the point is one of the people who was hired to actually develop the lab and actually put all the pieces together mentioned trying to incorporate Indigenous nomenclature into it. And it really challenged me individually because I thought it was totally a different realm of thought, right? I could not really wrap my brain around what this person was trying to say to me about we were talking about microfluidic chambers and molecules and about the spirit of that. And this other person was trying to talk about the spirit of these devices and how to convey that in an Indigenous context, which I think is what you're asking, right? And I found it, as a scientist, very confusing, personally, and challenging. And it took multiple conversations to sort of come at this discussion intelligently. Yeah.
Okay, just one question, just down there. Oh.
Thank you. I realize this is a science meeting, and some of us are social scientists, and we don't have linguists, for example, here, and I am not a linguist. But I'm struck by the languages we use. I recall a time, I've done some fieldwork in the Canadian Arctic, for example, and I realized after a full week of fieldwork that I asked 4 distinct questions in my mind, and the way they were translated in Inuktitut was always the same question, because there was only one word for all of that until my community partner pulled me aside and said, we just use pollution for all of that. We actually use the word bleach because that's our closest analog. And then I hear Patrick talking about his reflection on his language of exploiting nature, and Chantel's challenges with a grandmother who learned to speak English. And then you talk about bringing indigeneity to urban areas. Does your language now have to evolve to work with the scientific community and with this evolution of Indigenous knowledge and indigeneity itself? And if so, how? What guides that, or is it an organic process?
It's a really, it's a great question, and I don't really know is the answer. I think part of, so you're a geographer and I'm a geographer, so we understand each other, right? And geographers are pretty awesome because they seem to know a lot about a lot. I think we are like masters of all trades sometimes, or masters of, I don't even know how that works, so I'm going to stop now. But I think one of the greatest skills that I have is a social skill and ability to kind of talk to people regardless of where I am, which I can attribute to my mom, because she's my greatest teacher, but she is also very, like, she's like lights up the room. She's so bubbly and wonderful and knows how to talk to anybody and always makes friends. And this is, I think, one of the things that has guided me, is that I know what I don't know, and am not afraid to ask questions, but know that I have to ask them in a responsible and respectful way. And I think that guiding, being led by that assumption means that I won't be offensive. Because I think that, well, hopefully not offensive, but I think that the ways that Indigenous peoples are changing and reflecting the new situations that we're in is very tricky, right? And I mean, when we talk about what people are, who they are, how they identify, who belongs where, it's really a contested issue. Because, I mean, Indigenous identity is a very complicated thing, because it is a reflection of this silly political structure that has basically been imposed. So I think that people know who they are, and I think that you ask them to define what they think is important, and that's the best way to go about it. Does that answer your question?
Just, yes.
Mine is more a comment in listening to both the scientists and the social science aspects of it. One thing that has irritated me for 40 years as a biology professor is undergraduates that say things like plants, animals, and humans. And this is really one of the basic problems that we have, is in our educational system from a very early age, we don't teach our children that they are part of nature and they are an animal, whether they like it or not, in the true biological sense. And if we understood that a lot better, I think we would have a lot less problems and people would move forward on it in a much easier way if they accepted we are part of the system, and what we do to the system will bounce back at us, and so we need to respect it. I mean, we have to move forward, but we move forward in a way that takes into account the ecosystem. But it means education from grade K, you mentioned it, from kindergarten up, that we are part of it, we are not separate from it.
So I'm not going to say anything as profound as him. The first point, I just would like to point out to Patrick that in the book Gulliver's Travels, that in the part that people don't usually read because it's the 5th book or something, that Gulliver visits a research institute where they are trying to distill the essence of cucumbers to bring the sunlight to winter. And this was a direct satire of the Royal Society. So you can see you're working in a good historical context. I feel that Chantal's point about language is something that is far more generic than the problems of Canada, because there are a huge number of minority languages, I think about half the languages in the world, that are in the process of dying out. And you can see already Chantal is not a native speaker of her own language. And the problem is that there may be things that in the small number of languages we have left, French, German, English, Chinese, we may not be able to think about certain thoughts. If you imagine the situation reversed, that Chantal's language was an ascendancy and French and English was dying out, the whole concept of gender would disappear, which would make whole areas of science much more difficult to do. And I suspect, but I don't know, that in these indigenous languages, they will have words for certain things, say in animal behavior and so on, which we don't have words to differentiate. As you know, that in some languages they have more words for colors than in others. So Russians can distinguish between light blue and dark blue better than the English who it's all blue to. And so I just think that as well as looking at this problem from the point of view of the moral problem of the disadvantaged Indigenous people, which clearly has to be addressed, we should also look at it at the huge opportunity for everybody to get some of the thoughts from these languages so that science can move forward. Because I think if you also put on a positive spin, everybody can see the advantage, rather than putting on the moral spin, you've been dreadful, now you've got to do something, which will tend, because of human nature, to produce a backlash and may not achieve what we really want, and make the Indigenous people feel they have something to contribute towards the future, rather than just rectifying the injustices of the past.
To follow up on that, anyone?
So, catch?
I just wanted to follow up on Jeremy's point about being part of nature and what Chantal was saying. Like when my cookum is giving teachings, my grandmother, she says there's two-leggeds, there's four-leggeds, right? There's the ones that swim, right? And there's ones with wings. So I think that's a really important point. And it goes to the idea that the two-leggeds were the last ones put on the earth, so we're the least important. And it's sort of a good teaching that when we think about, she always says, you know, if you want to think about it, Mother Earth doesn't really need the two-leggeds. And so if you want to think about, you know, preserving Mother Earth and being kind to Mother Earth, remember that Mother Earth can survive without us, right? And so just being aware and being cautious of walking softly on Mother Earth, being kind to Mother Earth. And you think about an ecosystem, you know, thinking about we are part of Mother Earth, we're part of that ecosystem, and you know, that's how I'm trying to raise my children as well, that we are two-leggeds, but also Mother Earth doesn't need us. So I just wanted to add that language.
Okay.